Once more, I'm back with the third lecture on molecular basis of inheritance. Today we will go to learn about the different experiments. First the introduction, where after that explanation on the first experiment related to different aspects of inheritance. Search for genetic material. Many scientists started working in the search for genetic material. Some prominent ones, which we generally refer to. First, conducted by Frederick Griffith, was the transforming principle. He carried some experiments on mice which were infected by Streptococcus pneumonia, and then he put forward a principle which is referred to as the transforming principle. Then later, Oswald Avery, Colin McLeod, and Maclean McCarthy continued Frederick Griffith's experiment to exactly prove what was the transforming principle. So they tried to characterize the biochemical nature of the transforming principle. Then Alfred Hershey and Martha Chase, they worked on bacteriophages and proved that DNA is the genetic material. The final unequivocal proof was given by Hershey and Chase that DNA is the genetic material. So in this third lecture, we will discuss, or we will try to learn more about the experiments conducted by Frederick Griffith and biochemical characterization of transforming principle proposed by Griffith. These two experiments we will learn in the third lecture, and in the fourth one we will go to know about DNA as a genetic material, which was proved by Hershey and Chase using bacteriophages. Transforming principle, the experiment conducted by Frederick Griffith on mice. Mice which were infected with pneumonia caused by Streptococcus pneumonia, the bacterial strain. So Frederick Griffith conducted this experiment in 1928 with the strain of bacteria Streptococcus pneumoniae. Now, based on whether the Streptococcus pneumonia had a mucus polysaccharide coat, a mucus covering on its surface, based on the presence or absence of the coat, two strains of the bacteria were identified. One was named as R strain, the other was named as S strain. R standing for rough and S standing for smooth. So, presence of the polysaccharide coat meant that the outer surface of the bacteria Streptococcus will be shiny and smooth due to the presence of that polysaccharide surface. The surface was coated with the polysaccharide mucousy layer, due to which it was quite shiny and smooth. So such strains of Streptococcus were named as, or were identified as, S strain, and the ones in which it was absent were named as R strain. Then, carrying these two strains, the experiment was done. What Griffith first did was collected Streptococcus, both the strains R and S. First, what was done? The bacterial colonies, rough, non-virulent strain. That means the ones which did not have the mucus polysaccharide coat were taken. Those bacteria were taken, those types of Streptococcus were taken, and then they were injected into mice, the body of mice. The mouse, it was found to remain healthy. They were remaining quite healthy without any infection, without any symptoms of pneumonia. So results, the mouse healthy. So this rough R strain of bacteria, they were considered to be non-virulent. Non-virulent means non-infectious. So they did not transmit any infection. Next, smooth coated or the mucus coated bacteria were selected, were taken, and they were injected into mice. Result, the mouse died. The mouse, they die. They died. Why did they die? Because the strain, this particular strain, a strain of bacteria could infect the mice with pneumonia. They suffered, they showed symptoms, and finally they died. So these were considered to be the virulent or infectious forms. So Streptococcus, which means that Streptococcus has two strains. One was non-infectious, one was infectious. Next step, the smooth strains, which were actually infectious, they were taken. They were killed by application of very high heat. They were all killed and then they were injected into mouse. The mouse did not die. Whereas the living S-strain bacteria could infect the mice, but that S-strain bacteria could not infect the mice. Means that bacteria did not have any property of virulence, did not have any infectious agent remaining in it. That is why the mice were healthy. Then the last step, what was done? The non-virulent, which were already non-infectious, some of them were taken. Again, the heat-killed ones were taken, which were also non-virulent, which also turned non-virulent due to application of heat. These two strains were mixed together and then they were injected into mice. Mice died. Whereas when separately they were injected, the mice never died. But when mixed together, then the mice died. From the body of mice, the dead mice, what had actually entered into mice? So that was the big question. So from the body of the mice, the germs that were injected out, okay, that could be detected, the presence of Streptococcus that could be detected in the dead mice or in the dead mouse were living strain of S bacteria, smooth bacteria in the blood sample from the dead mouse. Now, the living S strain had never entered the mice. But after the mice died, what happened? Living S strain bacteria were injected out, were found to be present in it. Living S strain bacteria can infect mice. That means the rough living strain of bacteria were converted to or were transformed to virulent smooth forms. How did the transformation take place? The transformation, because independently the transformation never took place, here it was already seen. The transformation, that means what could be responsible for this transformation? Definitely the other strain, though they were dead, but they must have been responsible for transforming the R strain bacteria into virulent S form of bacteria. The only difference between the R and S strain was the presence or absence of the polysaccharide coat. That means even after the bacteria, S strain bacteria were killed by application of heat, in spite of that, something was present in it which after coming in contact with the rough non-virulent forms could transform the rough non-virulent forms to virulent S forms. That something which did this change, which brought about this change, was referred to as transforming principle by Griffith. So the conclusion that Griffith arrived at, heat-killed S-type caused transformation of the R-type bacteria into S virulent forms. And this transformation, this transformation that was taking place, it was referred to as the transforming principle, which was found to be quite significant in the search for genetic materials. But Frederick Griffith could not tell or could not exactly justify what is this transforming or what was this transforming principle. So his work was continued further by three scientists, who were Oswald Avery, Colin MacLeod, and Maclean McCarthy. They gave the biochemical characterization of the transforming principle. They said that what was present, okay, what happened actually, and what was responsible for carrying out the transformation was justified by these three scientists through a series of experiments. So first, what they took heat-killed as cells. Like Griffith, they also took heat-killed as cells directly, which were non-virulent. They were supposed to be non-virulent forms. They took, in the laboratory, and then first what they did was, by applying enzymes, lipases, amylases, by applying such enzymes, lipids and carbohydrates were removed. They were dissolved such that this bacteria will be devoid of lipid and carbohydrate content in the dead cells. It is devoid. So whatever remains back, mainly it will be protein and the nucleic acids, because it was assumed that the transforming principle, which was referred later as the genetic material, will be either protein or nucleic acids. So it was almost certain that lipids and carbohydrates are not acting as genetic material. So at the first step itself, they were removed. Then next, the sample, 3 parts of this sample was taken. To one part, proteinases were added, proteinases which will dissolve protein leaving behind the nucleic acids. So sample should contain no protein. Why no protein? Because proteinases were added which will dissolve the protein. Second sample, ribonucleases were added. The sample will not contain any RNA, but will contain protein and will contain DNA. Nucleic acids, 2 types, as we all know, RNA and DNA. So the second sample will contain protein as well as DNA, but without RNA, because ribonucleases were added. Again, the third sample from the original one, the third sample was added deoxyribonucleases. This sample will contain protein and will contain RNA, but no DNA, because deoxyribonucleases will dissolve all the DNA that was present. So in this manner, these 3 samples were taken. These 3 samples were then independently injected into mice. Okay, they were injected into mice. Independently they were injected and then, okay, and injected into mice, as also it was seen when the R cells were added. Okay, that means they were added with these samples. The living rough strain bacteria were added to the first sample which had no protein, which did not have any protein. It was mixed and then injected into mice. Injected into mice, or otherwise also when they were they were left as such, then it was seen that as cells appear. Okay, as cells appear. That means transformation occurs in this first case, which did not have protein. Second sample, which did not have RNA, but both of them had DNA. Both of them had DNA. The first one, DNA and RNA both. In the second one, protein and DNA both. So here, in both these cases, transformation was seen and the mice, they died. But in the third sample, when the sample was added, now this sample, what does it have? It has only protein and RNA. It does not have DNA. Our strains were mixed together, injected into mice. Mice did not die. Why the mice did not die? Because no transformation occurred. And when the sample was tested, it was found that no living as strain bacteria appeared there. Now, why this difference? The first 2 cases were similar, whereas the third case was different. Why? What was the difference? The only difference was that the third sample was devoid of DNA, but both of these 2 had DNA in common. The common factor in both the first and second was DNA. So definitely it must be DNA that is responsible for carrying out the transformation. So in this manner, Avery, MacLeod and McCarthy characterized the transforming principle of Griffith. Characterized the transforming principle of Griffith, saying that it is DNA that could carry out the transformation, that could transform the dead S-strain bacteria into that could transform living R-strain bacteria into virulent S-strain of bacteria. So finally, what they concluded? They concluded that DNA is the genetic material. The next lecture, that is the fourth lecture, we will learn more into details of the proof for genetic material. DNA is the genetic material. Though Avery, MacLeod and McCarthy had proved it, but it was not, a lot of criticisms were there and it was not fully accepted until Hershey and Chase had given the unequivocal proof. It was the proof, the experimental proof which they gave, that was finally accepted, and that through it was confirmed that the genetic material is DNA.